Section 3. Biological explanations. Genetic explanations. Genetic explanations suggest that there is a genetic predisposition to develop schizophrenia. Recent work in genome scanning has shown that there are hundreds of genes that are implicated in schizophrenia. However, advances have been made in this area, for example, the type 3 dopamine receptor gene, DRD3, and the type 2A serotonin receptor, HTR2A. There is a tendency for schizophrenia to run in families, and controlled genetic studies have shown that the risk for a particular individual developing schizophrenia increases with the degree of genetic relatedness. For example, Gottesman 1991 reviewed studies carried out since 1966 which showed that the concordance rate for MZ twins was about 50%, whereas for dizygotic twins, or children with one affected parent, the risk was 17%. The risk for general population is 1%. Evaluation. Concordance rates. Even when the relative is genetically identical, the chance of developing schizophrenia is well below 100%. This suggests that other environmental factors, for example hostile family relationships, play an important part in the development of schizophrenia. Sample size. MZ twins are relatively rare in the population, and of these, only 1% would be expected to have schizophrenia, so sample sizes in these studies are usually extremely small. This means that one must be cautious drawing conclusions about the causes of schizophrenia from twin studies. Differentiating MZ and DZ twins. In older studies, there was difficulty distinguishing between MZ and DZ twins without genetic testing, and given the broad range of symptoms of schizophrenia, a clear diagnosis was not always given. However, recent research from Cardno et al. 2002, based on the Maudsley Twin Register, which used strict diagnostic criteria for schizophrenia and accurate zygosity assessment, showed a concordance rate of only 26.5% for MZ twins and 0% for DZ twins. This provides some limited support for genetic predisposition. Difficult to separate genetic and environmental influences. Concordance rates in twin studies do not necessarily represent genetic causes. The increased probability could be due to environmental similarity of the twins. Therefore, adoption studies allow researchers to disentangle genetics from environmental factors by studying people born to schizophrenic parents but brought up by people who have no history of the disorder. Tienari, 1991, in the Finnish adoption study, identified 155 adopted children whose biological mothers had been diagnosed with schizophrenia. He compared them with a matched group of adopted children who had no family history of schizophrenia. He found that about 10% of the adopted group whose mothers had schizophrenia developed schizophrenia, compared to 1% of the second group. This study appears to provide strong evidence that there is a genetic component in the development of schizophrenia, but again shows that genetics do not provide a complete explanation. Biochemical explanations. Dopamine is a neurotransmitter found in the limbic center of the brain. The dopamine hypothesis suggests that schizophrenic symptoms result from elevated levels of dopamine activity at certain synaptic sites. This could be caused either by the release of excess dopamine by presynaptic neurons, an excess of dopamine receptors on receiving neurons resulting in more dopamine binding to these receptors, which triggers more neurons firing, oversensitivity of dopamine receptors. Dopamine neurons play a key role in guiding attention, so disturbances in this process may lead to problems with perception, attention, and thought characteristic of people with schizophrenia. This imbalance in dopamine levels could be inherited, and therefore related to genetic approach, for example the DRD3 gene, or the result of environmental factors. Evaluation. Support for dopamine hypothesis. Antipsychotic drugs. Called dopamine antagonists, for example phenothiazines, can reduce schizophrenic symptoms. They work by blocking the activity of dopamine in the brain, by reducing stimulation of the dopamine system. The effectiveness of these drugs strengthens the case for dopamine imbalance being a significant contributory factor to the disorder. Post-mortem examinations. Seaman, 1987, reviewed a number of studies which found increases in dopamine receptor density in the left amygdala between 60 and 100% compared to controls. Evaluation. Problems with the dopamine hypothesis. not all patients benefit from phenothiazines. These drugs tend to only work on positive symptoms and not the negative symptoms. However, different types of schizophrenia may have different causes. Clozapine, a drug that is one of the most clinically effective treatments for schizophrenia, binds to both dopamine receptors and serotonin receptors, which implies that the serotonin system may be important in schizophrenia as well. A weakness of post-mortem studies is that the examinations have usually been carried out on people who have taken antipsychotic drugs for years. Therefore, it is difficult to tell whether increased dopamine levels are the result of drug therapy, neuroadaptation, rather than the cause of schizophrenia. Cause and effect. Excess dopamine is only correlated with schizophrenia. It is not possible to say whether excess dopamine leads to schizophrenia or whether schizophrenia causes excess dopamine to be produced or makes the receptors more responsive. There may be an unknown variable which causes both schizophrenia and excess dopamine. Conclusion. The causes of schizophrenia are probably due to an interaction between biological and environmental factors. The diathesis stress model links genetic predisposition to schizophrenia with life events. In schizophrenia, a genetic vulnerability may lead to a biochemical abnormality with, for example, the dopamine system. This could lead to ability to process information abnormally. The addition of disrupted family communications could then be enough to trigger the development of schizophrenia.